Hi class, Mr. Roth here. Today's lesson is going to be on cell differentiation. You look at the image that I have here on this first slide, you're going to notice in the very center we see a bunch of cells that all more or less look the same. These cells are cells that have not yet specialized in their function, but we know that in our bodies we do in fact have different types of cells. You could start with muscle cells as an example. Red blood cells, very different in shape and function than muscle cells. Neurons, used for communication. Intestinal cells used for absorption. Pancreatic cells used for creating insulin. Liver cells. All of these cells have very unique functions and they look different from each other as well. So how is that possible? We're going to learn about that today. But first I want to ask you a question, and it's this: does every cell in your body have the same DNA? We already know that they can look very, very different from one another, and so many of you would be inclined to answer no. However, the correct answer is yes, they do have the exact same DNA. Every cell came from an original zygote through mitosis, which we know makes clones, so every cell has to be the same. So it begs the question, how then do these cells become different? Right? How do they differ so much in their structure and function? Well, the answer to this is that gene regulation happens. You can think of gene regulation as a big panel board full of switches. Every single cell in the body has the same genes. It's got this gene, this gene, this gene, this gene. But what can vary is which genes are on or off. These switches can be turned on or off at any time during the cell's life, and this is what happens to make the biggest difference in what they end up looking like, and consequently how they end up functioning. So how do these switches actually get turned on or off? Well, there are certain things in the environment that can trigger a switch to change, and thus a gene to be expressed. For example, the presence of this white molecule here seems to have an effect on releasing this repressor that's blocking the gene. So if we look at this cell right here, since there are no white molecules floating around, the repressor is happy to stay on and block the gene. However, in this scenario, this might be a muscle cell. We have the white molecule floating around, which when it binds to the repressor causes the repressor to pop off the gene, allowing for the RNA polymerase to move forward in this direction and cause that gene to be expressed. So in this cell up here, the switch is off, much like this switch here, and however for this cell the switch would be on, like this. And so what is the effect of this? Well, I want to talk for a second about stem cells. You can think of stem cells as cells that do not know what they're going to become. Where do they come from? Well, originally, back early on in our development, we are at one point a 4-celled organism. We call this a morula. But pretty soon we start to develop this inner cell mass here in the blastocyst. And these are the cells that are what we call undifferentiated. It means that they don't know what they're going to become. And in some sense, all of their genes are on for the time being. But pretty soon they're going to receive a very special signal that's going to tell them what to become, because a blastocyst is not going to be able to turn into a human being unless we can get every type of cell that we need to build a human being. And so these cells will soon receive very special chemical signals, much like we showed here, that are going to turn on genes. Once those genes are turned on, for example, the genes that code for actin and myosin, very important proteins in muscle cells, you turn those genes on and we're going to get a muscle cell to develop. If we turn on genes like hemoglobin that are very important in the structure and function of a blood cell, then we could get a blood cell. So it's the regulation of these genes that is going to allow the cells to differentiate. When you think of the word differentiated, I'd like you to just think of the word different. and really it's what allows stem cells to become different from one another. That's it, folks. I hope you enjoyed the lesson. We'll see you tomorrow.